Today I'm going to be talking, and I'm going to be starting my talk with describing to you like where this whole project started, because it didn't start at all where it ended. And so it was a long time ago when I started my PhD in Ethan's lab, and my main interest was understanding the beauty of evolution and the many shapes that you can find in nature. And this is a picture of a lot of different wings of dipterans, the true flies. And from all these very beautiful wings, I decided to work with the wing of Drosophila melanogaster, which is the one that infects your rotten fruit at home, and another wing from Megacelia abdita, which in fact infests your trash at home. So probably not a great choice for the smell in the lab, but a great choice for the developmental models that were available at the time. So in this two species, what I was looking at specifically is how a specific vein structure, let me point it out, that vein L2, this structure is determined. And we were trying to use this novel technology at the time, CRISPR, to generate mutation and try to take that vein out in these two species. So now there is a gene that determines the specific location of that vein. And when you have two copies that are good of this gene, you have a perfect vein. It looks exactly like that. Now when you mutate and cut out that gene, you have two mutant copies on both chromosomes that flies have. You get complete lack of that gene and complete lack of the vein structure. So it was something that has been done in Drosophila. In Drosophila there is like, I don't know, 120 years of genetics. So it was something that there is a lot of tools for, but it was very hard in these other species that I was working on, this Megacelia. And what we were trying to do is generate a mutation using CRISPR, but the fact is that when you hit only one chromosome, the other copy is good enough to make the vein. And so it's very hard to screen and to follow. So we were trying to develop a tool that would convert the other chromosome to the same condition and therefore show the phenotype right away. So something that would make this process much easier and much faster. And that's why how we came up with the idea of the mutagenic chain reaction, which is based on a very simple concept and the use of the two CRISPR components, the Cas9 and the gRNA. So these two components are a protein or an enzyme that goes in and is capable of cutting the DNA, and a guide RNA that guides the enzyme at a specific location. So this can be coded and engineered to cut wherever you want into the genome. And that's, for example, in this scheme, like the location at which it cuts. So how these two components work is that they come together, they cut the DNA at that location, and now if you add two homology sequences of DNA in a specific location, you can trick the cell, so this is the genome of the cell, to use this artificial piece of DNA to copy that information in and fix it in that way. So the beauty of this is not what happens at the first step, but what happens at the second step. And this is what happens on the second chromosome of the cell. So the cell has two chromosomes, and once this construct is inserted at a specific location, now the same exact process happens in an autocatalytic manner, and now the same mutation gets transferred onto the other chromosome. We decided to test in Drosophila, obviously that's our model of choice. And we decided to use this specific mutation, which is called yellow. It's a traditional mutant of Drosophila. When you have two mutant copies, the flies look yellow. Like, I don't know if you guys can see it. Seems very similar from here. But the wild types are darker compared to the yellow ones. Now, when you cross these two flies, you obtain heterozygous, so one good copy, one bad copy. And these heterozygous progeny all look dark just because they have one good copy that makes the pigment. And so the one good copy is enough to make them look dark. Now, when we tested the same exact cross done now with an MCR allele, so an insertion into that gene that disrupts it, the same gene yellow, we saw that in the progeny we observed this conversion, and therefore we saw that we had almost 100% conversion to that specific phenotype. And that was something that threw Ethan off the rail. He was super excited about this because it was the first time that he saw in his life a genetic cross not working like it should do. And so this was all great, and this satisfied what we were looking for, so a system that would allow us to see a phenotype right away just by having one single insertion. But we didn't know at this time whether it had chromosomal conversion or the construct, the Cas9 construct that we had, was just hitting the other chromosome and modifying with a random mutation, which is also a possibility. But fortunately for us, it was not just on the somatic cells, so the cells of the body, that we had this yellow pigment and we got this conversion, but also inside the germline of these individuals, we had this conversion. And what this means is that you're completely changing the ratios at which these individuals give off the genetic modified element to the progeny. In a regular cross, you would have that, in regular Mendelian genetics, this individual would give off 50% the modified and 50% the wild type. But instead, in these animals, since you get conversion, now you don't have the wild type anymore, and you give off 100% of the time the modified one. So this results in a strikingly different inheritance, in which, due to this conversion, you have that instead of inheriting 50% from these animals, 50% the yellow, 50% the dark one, you always have the yellow one. And by generation after generation, you get this amplification in a population that you can basically spread a wanted characteristic into a whole population, or you could use this system to basically bypass the constraints of Mendelian genetics. And we're going to go over the possible application of this kind of technology, but we got a lot of interest based on the potential of these applications that resulted in the past year or so to a great inflow of funding into UCSD, really focused to this kind of technology. And we went from going into the fruit fly into the obvious partner mosquito, in which there is a very, very obvious application of this technology for population modification that Ethan's going to talk to you about, in collaboration with Tony James. And as well, we took this, we didn't take it yet, but we're going to take it very soon, hopefully, this technology into many other systems because we have recognized the potential application both at the level of research and commercial if we bring this kind of system into other model organisms. So these are not just the people that are working on, but there is many more, and some of them are actually all sitting back there. And so you can see like this ever-growing group of people working on this technology here at UCSD. Lastly, I'm going to go over the potential application of this technology, and these are like few examples that we have thought of that could really benefit of this kind of new genetic inheritance. And one of them is genetically modifying mosquitoes with the purpose of spreading a genetic characteristic of interest into a whole population. This is, again, what Ethan will tell you about, and how this technology could be used to like spread into an entire population a malaria resistance cassette that would prevent mosquitoes from carrying malaria, and therefore could potentially stop the disease in endemic countries. The second is using this technology to dramatically modify how the genetics work in model organisms, like research model organisms such as mouse and rats, in which currently, by means of regular Mendelian genetics, you can assemble in a single animal only 3, 4, in some rare cases 6 different modifications. But past that, like cost of assembling complex genetic mice becomes prohibitive. So active genetics could really cut down the time in half of these kind of processes. For the same exact reason that this could help in the mouse, if applied to plants, the same technology could also speed up genetics in plants. But additionally, some plants do have not only 2 chromosomes, but in some cases they have 4, 6, or more. And in those plants, people just don't do genetics because it becomes an absolute nightmare. Since this kind of genetic element is capable of copying itself onto the other chromosome, it is possible to think that it could copy itself on all of the chromosomes, and therefore would make genetics viable also in these plants. Lastly, the process of copying from one chromosome to the other could also be applied into modifying cells of the body, and therefore could have an impact in cell therapy applications that more recently have picked up a good track record of good applications for human therapy. But we were also thinking that in the future, this kind of technology could also be used to like repair things such as broken muscles or skin or other more minor diseases. And currently, these 2 applications are now brought to at the commercial level by Symbol, and agricultural application, as well as genetically modified mosquitoes by Agrogene. But now I'm going to be talking a little bit more about the mosquito portion and showing you, like, in brief, before I hand it off to Ethan, how the conversion from one chromosome to the other biases this kind of inheritance. And so now a mosquito that has 1 copy actually gives it off to all their progeny. And so their progeny will do the same until you can get a complete takeover of the population of a specific genetic modification. And this happens in really an exponential way, so that in as few as 10 generations, you could be able to convert a whole population of mosquitoes, a wild population of mosquitoes, to have a specific wanted characteristic. And this kind of tool has really been looked for since the 50s. And people have been trying everything, but never gotten this kind of conversion that we have observed in Drosophila. And this was really like a holy grail for these people. And when we handed off our data to Tony, he had exactly that face, our collaborator. And so I'm going to hand it off to Ethan, that's going to tell you a little bit more about this technology and how the TIGS is planning to apply it in India.
Okay. Well, thanks, Valentino. That was very good. Oh, yes, right. That's an excellent introduction of the whole thing. And, you know, I should say it's been one of the great privileges I've ever had to have Valentino working first as a graduate student in the lab and now as a colleague. It's just been one of those dreams that one has. And when I teach, I often make a point to the students what a large contributions to science graduate students have made. Hilda Mongold was Hans Spemann's famous graduate student who identified the organizer. And there have been many examples, including some UCSD faculty like Bill Harris, when he was here previously, had done some of the fundamental works as a graduate student as well. And, you know, to see those few but very important examples, and then have the wonderful experience of having such an amazing person in the lab was a great privilege. And now, of course, he's he's an independent PI and has his own program going. It's been a pleasure to watch. So One of the things that I asked Valentino right away when he had this idea of coming up with this little genetic widget that could self-copy that MCR element was, well, what kind of applications might be suitable for this? And he looked around in the literature and he realized that, you know, one of the big problems that we hadn't given much thought to was the issue of a gene drive, how you could spread a characteristic in a population, and how that was, as he says, the holy grail that people had been looking for in malaria, because Chris Curtis back in the 60s had come up with this idea that you could use balanced chromosome translocations, which bias inheritance in a certain way, to spread a desired characteristic in a population. And the question was, well, whether there would be some kind of way of spreading a desired characteristic, and that desired characteristic in the case of malaria would be to prevent the spread of malaria, using such a genetic strategy. And so, as Valentino pointed out, people have been trying for many decades to come up with such a thing, and there have been intermediate successes that would work at a particular location in the genome, but it wasn't programmable. The so-called homing endonuclease genes were an example that led to that. But until Valentino demonstrated that this would work as it did in fruit flies, there hadn't been such a tool that had that sort of general applicability. And so we called up Tony James at UCI, and this is largely what I'm going to tell you about the work of his lifetime and our, you know, relatively recent entry into it. But he was also, like us, originally a fly biologist, and decided at an early point in his career that he wanted to devote it to the elimination of malaria. And so, as one of the things, first steps in that, he's been a very systematic in his career. I can't say the same about my own, but the first step was to introduce transgenesis in mosquitoes, so he was the first to do that. And then he spent a very long time devising a genetic element that I'll just refer to very quickly as an immunizing element here, but a very sophisticated genetic element that allows for the very localized activation of a genetic element that immunizes the mosquito against malaria by producing some antibodies in it that bind to the parasite and stop the parasite from transmitting. It all sounds very simple when said in those 2 or 3 words, but it was about 10 years worth of work to build this intricate genetic circuit that could accomplish that. So anyway, that's the kind of general context into which this fell, but I'll just give you a little bit of background into what the problem is of malaria. And it's an infectious disease that's caused by a parasite that is called Plasmodium parasites. And there are a couple different species that I'll mention. And it's borne by mosquitoes. So what happens is that a mosquito will bite a person that has malaria and get blood from them that has the parasite, and then the mosquito will have the parasite, and the mosquito will bite another person and transmit that parasite to the person. And there's a high degree of specificity among mosquitoes and parasites for the host. So there's certain mosquitoes that transmit parasites to humans, others to rodents, others to birds, and they're highly specific. And that's a good thing from the malaria eradication profile. For example, other diseases like Chagas disease, which are transmitted by sandflies, and sadly are not so host-specific. So the sandfly can bite any mammal and transmit the parasite. In that case, it's a different kind, but it's a trypanosome parasite, but it can transmit it to all those different species. And so that's a much, much more difficult problem, obviously, to face than one that is so uniquely specific. And the other thing that's important is that there are no free-living forms of the parasite. So if you can block transmission of the parasite, if you can interrupt that transmission cycle, mosquito biting infected person, biting uninfected person and transmitting it, then you have, in principle, an opportunity to to block the transmission cycle. And again, that would be much more difficult in an organism that was free-living, had a free-living stage, or an organism that wasn't so host-specific. So, this is actually from Tony's going through the long historical literature. Among other things, he's a scholar. But like many other deadly diseases, including cholera and smallpox, malaria has an ancient history, and there are texts dating back to Mesopotamia and the Vedic texts in India, as well as Chinese texts that make allusions to malaria, which, malaria means bad air, so people understood there was some kind of connection, I guess, with the air. And Hippocrates also understood that swampy waters had led to these cycles. So that was kind of the classic designation, or understanding that there was malaria it would be kind of a cyclic disease. And then recently, the thing that is of course going on everywhere is people are digging up cadavers from the ice ages and so forth, and other times mummies and so forth, and trying to figure out what did them in. And there's evidence that, in Rome, for example, that some youngster perished sadly from malaria. And I should say that malaria is a disease that kills about 500,000 people a year annually. It disproportionately affects the developing world, and it disproportionately affects children. So there are several different forms of the parasite, and as I mentioned, they're host-specific. These are all ones that are of relevance to humans, but the ones that are of the greatest concern are the two on the left, falciparum and vivax. And in India, the country that we're most focused on at the moment, although there's also an ongoing effort to help Tony in his efforts in Africa, are these two. And in Africa, the dominant one is the falciparum one. So in principle, there are, in certain countries like Africa, there are about a handful of 6 species of mosquitoes that are the major vectors, and they typically have those two parasites, falciparum and vivax, that are of the greatest concern. Although I understand that these others are becoming more of a problem and perhaps adapting in ways that will lead to their becoming an issue as well. But at least in certain parts of the world, Africa and India being examples, the number of mosquitoes that carry the disease and the number of parasites that are the effectors of it are small enough that it's in principle handleable. There are other parts in the world like Indonesia where there are as many as 60 types of mosquitoes that carry it, so there the problem would be a much thornier one. So one has to consider that when one thinks of going from local elimination of the disease, which we'll talk about in a moment, to global eradication, which was successful, for example, in the case of smallpox. So it is possible to take a terrifying disease. And smallpox killed about the same number of people, 50 years ago. So it is possible, in principle, to eradicate a terrible disease, polio as well. So how does this work? Again, it's a little cartoon that Valentino has skillfully put together here that just explains, again, this idea of the mosquito biting an infected person and then transferring the parasite to them. And again, there are some 60 that can transmit malaria. But again, in the areas where there's some of the greatest concern, Africa and India, the number of species is actually more restricted. So, but it's something to keep in mind that on a world scale, it's a bigger problem. So you can ask. I suppose you already know more or less where it is that malaria is the biggest problem. It's mostly in areas in the sort of tropical belt, and again, there have been many waves, many attempts to eradicate malaria, and some have been successful even on continental scales. There was a time when in the use of DDT when malaria was almost eliminated from India, but it didn't quite get eliminated, and that's the problem. If you let it linger, then it can come back with a vengeance. And so what does it mean to take a map like this, which has been progressively bettered, and the Gates Foundation has initiated a very big anti-malarial program over the last several years, among others, and the World Health Organization, and they have, according to their numbers, reduced the incidence of malaria by about 50%, which is an enormous accomplishment, and I'll get to some of the elements of that in a moment, but still, it persists as a major problem, and is estimated, again, as I mentioned, to kill about 500,000 people a year. And so what would you do in principle to go towards eradication? And by eradication, I mean there is never another case of malaria in the world. What would one have to do that? Well, there's a sequence of steps that need to be taken, and again, the World Health Organization has defined those in terms of, first, you dramatically reduce the number of cases to a point where it's controlled, where there aren't very many, and where certainly the existing medical facilities can cope with them and treat them and so forth. So it's reducing them to a small number where, in the case of, for example, of smallpox, when they got it to the level of control, they were one step from local elimination because they then had enough information to completely wipe it out in a given area. In the case of malaria, it's probably unlikely that it'll be so simple because the transmission isn't just from one person to another, but goes through a vector, which complicates things. But you get to a point where you have control, and then in a given area, and it can be a small area or even a large area like India, you can get to the point where you could remove it from that entire area. And again, India is a pretty big example for elimination, but you could imagine it from a smaller country or an island. You could get rid of it from there. But then, of course, if you've eliminated it in one place, but it still exists someplace else, then it could come back in on a ship or a plane or something else. And so the next step, of course, is to lead it to eradication. And what eradication means is you eliminate it, not just in one place, but everywhere. And that is a very, very difficult step to accomplish. But in principle, if you have the tools for local elimination and elimination on a scale of, say, a continent, then you should have the blueprint for how to extend that to eradication. And that was successful in the case of smallpox. And in fact, one of the thorniest and most difficult places to eradicate malaria, smallpox, the last place was India. And at first it seemed that the strategies that were working in Africa were not going to work in India, but by bearing down on it and using all the wits that they had, they were able to make that last important step to eradication. And I think the same thing in principle would ultimately hold for malaria, if technologies such as gene drives and others, perhaps in combination with it, were used, in that there would be areas where one could do it relatively effectively and quickly, and then there would be others that are much more recalcitrant. Like, for example, Indonesia would be undoubtedly a significant challenge. So how do you get there? Well, you use a combination of things. The gene drive technologies that Valentino talked about. You have bed nets, insecticide-impregnated bed nets. That's a really big one. Residual indoor spraying. These are things that have been working very well. But the problem is that you get resistance to those insecticides that pops up. You also just have hygiene, getting rid of the standing water, and then there are a variety of drugs. The quinine is the old example of it. The artemisinins are the more recent, and concoctions of those together can be very effective, and those all together have, again, these sort of technologies have, in aggregate, reduced worldwide malaria by about 50%. And so the hope is that the addition of gene drives and other tools like Wolbachia and other systems that people are considering will, together with the existing tools, certainly not supplanting them, will come together. And that sort of brings you to the question of, well, why is malaria still here? Why have those current tools not worked? Well, one problem, of course, is a a lot of these tools are intensive in particular locations, and so you have to have penetration to the whole population, and some of these tools are very difficult to apply to all geographical areas. There's political instability in certain areas that also makes that very difficult. Also, if you take bed nets and indoor residual spraying as an example, they can work for a while, but then mosquitoes evolve, annoying characteristics, like instead of biting people while they're sleeping, they start biting them during the day outside, and so then those tools are rendered completely useless for those kinds of things. And then, of course, a huge problem is insecticide resistance. I mean, there's certain strains of mosquitoes that are resistant to 4 different kinds of insecticides, and so that's a really big problem. And I'm not going to touch on that today because of the generality of the pitch of the discussion, but Raja, who's sitting back there in the audience, is somebody who's working on applying the gene drive technology not only to spreading anti-malarial cassettes that immunize the mosquito against malaria, but also potentially helping to reverse insecticide resistance. So again, what are we doing about it? And the essence of it is to use a genetic approach to prevent the transmission of malaria. So the mosquito bites a person that has malaria, it gets the parasite in it, but it doesn't transmit it to the next person. That's the basic idea, and to have it be a highly species-specific, cost-effective system that will cause the least amount of ecological perturbation possible. That's kind of the goal. And again, I don't want to go into the details of it, which is doing Tony a great injustice, but the essence of how it works is that you have a vaccine region in the mosquito that carries, for example, these antibodies that bind to the parasite and immobilize it. So it's the genetic information for encoding those immunizing factors. And then a controller region that activates these antibodies at the appropriate time and in the appropriate place. So you don't necessarily want the mosquito flying around all the time with these antibodies against the parasite, and there could be some negative consequences of that. So it's only active in the female mosquitoes, which are the only mosquitoes that bite, and it's only active once they've had a blood meal. And again, that was a very sophisticated little genetic circuitry that's just nicely illustrated there in those 2 blue and red segments of DNA. 10 years of work. And as Valentino pointed out, the essence of it is this so-called gene drive feature of his element, which he already kind of went over nicely in his introduction. But if you look on the left, what you see is a blue mosquito that mates with a gray one. And then all the other mosquitoes in the environment, according to this, are gray, and you look at how the trait would get carried down, and you can see that it doesn't increase in number as it goes, because it's just keeps mating to gray ones, and the trait just gets passed down 50% of the time to one offspring or another, so it gets diluted out after a while as a consequence of all that. But if you look at the gene drive, what happens is every time it mates, all the kids get it, and all the kids' grandkids get it, and all the grandkids get it after that, great-grandkids get it. And so, again, as Valentino said, in 10 generations, in principle, through exponential, or actually over the full period of its growth, logistic growth of spread of this element, you can go from roughly 1% to 100% in 10 generations, which is roughly one season in the wild. And just to put this in perspective, a very well-known form of insect control is using sterile insects. So people have controlled medflies, for example, in California with this for over 50 years. And the idea there is you take on the order of 10 times as many males as are normally present in the wild, you sterilize them with x-rays and you throw them out, and so that they mate with the females, because there's way more of them than there are the normal females. And then, you temporarily depress the population, because those females are mating with sterile and, males that aren't being able to give rise to progeny. But that only lasts for one season. You have to do it over and over again. And if you look at the ratios, that is 10 to 1 relative to wild type, and we're talking about being able to do it at 1/100th to 1. So it's using 1/1000th the number of mosquitoes, adding that small percentage. So it would be imperceptible to people in the environment. So, for example, with the sterile insect technologies, even if you release male mosquitoes that don't bite, and this is true, for example, for those that carry Zika and dengue, it annoys people when you release all those males, because what they do is they swarm. That's how they mate. The males swarm, and then the females go into the swarm. So you'll be sitting there out on your porch, and you get these swarms of male mosquitoes coming around you, and then it's annoying, first of all. And then, of course, a female will see that, and she'll slither into that group, and then you might get bitten. So, you know, they're just releasing males, but it's annoying. Now, not all mosquitoes do that same kind of swarming around humans, and the ones that carry malaria actually don't. But it's just an example of how fundamentally different this type of technology is. So how would you implement this in practice? Well, what Tony has done in collaboration and over, again, a period of 20 years with the World Health Organization is come up with a phased release or testing and release process where at every step there is a go, no-go decision that says, you know, up to this point is the entity that you're using, a genetically modified mosquito, is it performing appropriately? Is it safe? And then move on to the next phase. And if the answer is no, then you go back to the drawing board. And then you redo it until you get to the point where you can satisfy that phase transition, go, no-go decision. And so in the first one, it's laboratory studies, which, you know, I'd say we're kind of at the end of that point. We're beginning to transition from that to the next point, but we're still in that phase, late in that phase. And then there's phase 2, where you take a mosquito that looks like it's doing pretty darn well in the lab, and then you test it in a contained way. And there are 2 kinds of ways in principle that can be done. You could imagine a very large cage outside so that you have the densities of mosquitoes that are normal instead of what they would be in a lab, which is typically 1,000 times or so more than they would be in nature. Or preferably you do it on something like an island, which is a segregated piece of land where, you know, you can make sure that the mosquitoes aren't going to be able to get there from there to anywhere else, where they're controllable by pesticides, because you start off with a mosquito that's pesticide sensitive. And if they perform well under those circumstances, then you can proceed to phase 3, which is to actually, again, this, assumes another point that I'll bring up, which is that at every step you're consulting lots of different people, and I'll get to that in a moment. But assuming that all is positive, you can then go to the point where you can actually begin to test it, release it and see if you can have an impact on the disease. And then if so, then there's a period of monitoring and assurance that, the application has been effective. And so throughout this whole process, beginning at the very beginning, and we're, very seriously engaged in these kinds of discussions already, is you look at issues of regulation, community engagement. Is this something that people would want to see done in their communities? What are the negative possible risks? Basically the risk-benefit analysis, and being open and transparent so that there isn't a, surprise that is foisted upon the public in the process. So this is kind of the responsible way of implementing a new technology like this that, again, Tony and many other of his collaborators have worked very hard for over 2 decades to formulate. And this is being revised now to some degree in the context specifically of the sort of gene drive systems that Valentino described. And so I'd just like to end by talking very briefly here about TIGS. We're fortunate that Suresh Subramani is in the audience. He's the global director. I don't know, Suresh, if you want to flag yourself there a bit, but he's the person who's in charge of the whole operation, which has got a component in India, a component here. The component here is charged largely with the sort of building of constructs, but more generally, and then there's one in India where, of course, if India is the place where this might eventually be applied, because malaria is a big problem there, then that whole process will have to take place there. And there's then the training that happens here at UCSD of the Indian scientists to put them in the position where they can make the decisions that they need to in India and carry forth the project. But overall, the mandate of TIGS is, number one, of course, to go after vector-borne diseases, and that's the primary focus of it. But in addition, as Valentino summarized, there are many other possible applications of active genetics, and although it's not the major focus of the TIGS effort, there are also mandates to develop the proof of principle, at any rate, in these various categories that Valentino's already glanced over: accelerating agricultural research, cancer therapy. Something he didn't mention, but is also another possibility in principle, is, you know, the whole CRISPR-Cas system comes from bacteria as a bacterial immunity system. So the question is, can you turn that back against the bacteria? And in fact, many people, I mean, people in the CRISPR field are no idiots, have you know, tried to do that, are trying to do that, but they're doing it in a kind of standard where they use CRISPR as a pair of genetic scissors just to cut things like antibiotic resistance genes and so forth. The question we're thinking about is whether you can use active genetic self-propagating elements to make that process more efficient. And so that's something we're looking at. And of course, number 5 is extremely important too, which is that we have to look at the ethics, the public policy implications of active genetics, the economic impacts it might have. And these are all things that are being done collaboratively, or will be done collaboratively, between UCSD and India. So I'll stop there, and maybe Valentino and I can take some questions together.
Thank you very much, Ethan, for this, and Valentino for the presentation. Could you go back to your last slide, please, for me, that you had? So I feel like he just gave me a teaser, you know? He really didn't tell me everything that he can do with this technology. So he talked about malaria, which is very important, and we understand how this can be used to eradicate malaria. But when I look at this slide, I mean, I see agricultural research, cancer, cell therapy, antibiotic resistance, economic sector, and public policy. Obviously, a lot of people are going to have to be involved in making this happen. And I think something that I would like to ask maybe Valentino to talk about a little bit briefly is about the people. The people that are involved in taking these technologies, these inventions, and making them impact the world. So can you tell us just a little bit about the team that you're forming and the background of the people that you're working with, and how you are going to hopefully push 1 or 2 of these innovations into the marketplace, and then, you know, what else can we do after that?
Well, my team is actually very small right now. It's only 2 and they're back there. But together with Ethan and actually our collaborators, in the slide that I put up earlier, like, the group is really growing exponentially, almost like an MCR. So we're really pushing the technology in a lot of directions.
What business people do you have that are helping you create or deploy the technology? Or are you partnering with someone in a business sense, or are you?
Also, like, other than UCSD, we're working on building 2 different companies that I mentioned briefly, Symbol and Agrigene, that are trying to bring to the commercial point 2 different aspects of the technology. One is more applied to therapy and tools for research that would benefit the research for therapeutical application, and also apply to our furry friends, so therapy for cats, dogs, and our companions. And the second company, Agrigene, is more focused on agricultural applications, insect suppression or modification, what Ethan has talked about, and also a similar use of the technology to target insect pests that affect crops.
All right. I'm going to hand it back to you guys. Who has any questions for Ethan and Valentino? Here we go.
How are mutations affected from this gene propagation technique? Like, are any other expressive factors altered from this active genetic therapy?
We have not checked yet, but CRISPR-Cas9 is a system that does hit the DNA at sometimes at has off-target effects, and that's something that we're definitely looking into. And depending on the application, that could be a bigger problem or a small problem. Like in the case of modification of large populations, if 1 or 2 mosquitoes die, you're not going to worry about it. But if you use it for therapy, it's something you need to take very seriously.
I'm from India, and whenever we go to India, we always are scared of malaria, catching malaria, or even dengue. So I had a question about the dengue, which is also spreaded by mosquitoes. So can this same therapy or same technique be used for multiple diseases spread by mosquitoes?
No, absolutely. And so in the case of the viruses like dengue, Zika, chikungunya, yellow fever, they're carried by a mosquito that's in a different genus. It's the Aedes mosquitoes, Aedes aegypti, albopictus, that carry those viruses. And we are actually, the two of us, part of a UC-wide collaborative team that also includes Tony James people at UC Berkeley, UC Riverside, UC Davis, and thanks, Valentino, and and UC Santa Barbara, that aims to test similar genetic elements in Aedes aegypti. And we've got a junior faculty member, Omar Akbari, who's coming, who's heading that. In principle, it's the same kind of logic. It's a little bit different, though, going after a virus than going after a parasite. The tools to take care of viruses at the moment are not ones that would be terribly suitable for gene drives, but there are new tools on the horizon that would be. So there are these new CRISPR tools that work by cleaving RNAs. These viruses are all RNA viruses. And so I think with a little bit more work, Tony took 10 years to make his widget. We might take a few to adapt the new technology to that. But in principle, absolutely. And it could be applied to Chagas disease. It could be applied to the leishmanias, also terrifying, horrible diseases. Those are more difficult because there's less background knowledge, and the parasites go straight through the gut instead of going into the salivary gland. So actually, when it has to meander through the body, it makes it easier to stop it. When it goes straight through the gut, it makes it more difficult. But in principle, the same logic should apply there too. It should be possible. And there are people who are making single-chain antibodies, these neutralizing agents to those parasites as well. And so they can be, in principle, put on gene drives. And we have a collaborator in Brazil who's trying to do that for Chagas disease. So there are people who are interested in doing it.
People have more questions up here?
What can you tell us about the status of intellectual property associated with this invention discovery, and how would that relate to any IP on the CRISPR system itself?
Great question for Donna. As far as the understanding of scientists goes, there is like a big fight over CRISPR, and it might take a long time to resolve. So we could operate until that resolves. But the better answer to that question is that there is other CRISPR variants that have been discovered that are free to use or on a non-exclusive license. And so some of these systems could be adapted to use separate CRISPRs. And there is also a huge amount of researchers who are like basically mining bacteria to find novel CRISPRs that do not obey or that do not fall under the original IP. And so personally, I don't see that as a big problem. Currently, the IP associated with this specific technology and these applications have been licensed to the two companies that I mentioned.
There are several.
Yeah.
Several patents.
There's several patents that cover specific aspects of the technology and the application.
Okay, I'm Jerry Feidelson from AgriBody. There was also an AgriGenetics that sort of sounds a little bit like your new company. I have two questions. One is technical, the other is social. The technical one has to do with the durability of the antibody to bind to the mosquito receptor to essentially make it incapable of carrying the parasite. I'm sure that the durability of that targeting has been looked at, but when you're dealing with hundreds of millions of organisms, whether they're bacteria in a petri dish or flies in the wild, you'd imagine that the fly would, or the parasite would evolve around that targeting. So what's the denominator, 1 over how many that's been tested so far to guarantee that you won't have the same problem as insecticide resistance?
Right. No, that's an excellent question, and it's one of significant concern to us. So, for example, at the moment, the tools that Tony has designed are 100% effective at stopping parasite transmission at the levels that they currently exist in the wild, in nature. And if you then increase the parasite load, which causes a fitness cost to the mosquito, which is why typically it's not that way in nature. But if you increase the parasite load, then the antibodies require to be expressed more than once. So they're almost effective, but they're not quite effective yet. So that's one answer, which is that we just need to get additional parasite effectors and express them maybe at higher levels, to make sure that we create a ceiling for which the parasite simply cannot get out of the situation as it is, just by increasing their numbers. Now, could they gain access, could they mutate their receptors, their targets, their antigens, so that they could no longer be bound by the antibodies? The answer for one of them is no, because it's a tetrapeptide that is present in all of the different parasites. So there's no genetic variation in that particular tetrapeptide. It's essential for parasite function. The other one, yeah, it's possible that they could evolve. And so one of the ideas that we have is to create a whole new set of effector molecules and to release them on genetic updating elements, which we haven't had a chance to talk about, that can be, once you have a gene drive in a population, that you can use to rapidly update it. So these are questions that we're thinking about, and you're right, they're of importance. The thing I would emphasize, though, is that the timescale of evolutionary evasion of the parasite is typically going to be on the time course of a few years. And the time course in which a genetic element like this can spread into a population and reduce the level of parasite to the point where it can no longer transmit. You don't have to have 100% blockage to do that for a system that requires this sort of leapfrogging. It could be 1 season. So if we release the right element, it could take them out before they have a chance to have the time to evolve. And that's what we're hoping to be able to do.
It just seems like the antibiotics research area is very analogous to this, where you target two different targets at the same time, and that goes back to Demarek in 1948.
Absolutely, and in fact, again, this was simplified here. The existing cassette that Tony has 2 different single chains in it already, and we're generating single chains to another 8 different targets.
All right, the other is social, also involves millions of organisms, but they're people. And that's the fear of GMOs in general, something I deal with every day in my business. And there's sort of an ick factor about GMOs and mosquitoes. People hate both. And so there's sort of a squared factor, even though you're using it against them. That's a level of technical sophistication that's not very common. And there's the Oxitec example in Florida and many others where they may have been a little bit arrogant about it, sort of pulled a Monsanto, so to speak, which you'll probably avoid. But still, the level of public knowledge of this is so incredibly low that it's just a real challenge to educate before you could release.
No, that's a really good point, and that's why there's such an emphasis on public engagement. You're right. I mean, Oxitec did it wrong. We want to try to do it right, and it's the issue of, how do you communicate something that's kind of complicated to in some cases, people in the actual target area that aren't even aware of Mendelian genetics and those rules. They kind of have a sense of heredity, but they don't even know what those rules are, and explain like a gene drive to them, and yet, without that knowledge, nonetheless adequately explain to them in ethical terms what the idea is that you're trying to do. And it's kind of analogous maybe to people who went out and tried to do vaccines where, again, the technology isn't completely understood but by the public, but you can provide an ethical assessment of the risk and benefits that would come from it. In this case, you have the added problem of the anti-GMO fear. Most of the anti-GMO fear, I think, is targeted to plants that people eat and food supplies. People are a little bit less worried about it. For example, cotton, GMO cotton is approved in India. So this is going after something that people don't like, mosquitoes. So the idea that you use a genetic modification against a mosquito might not be the square, it might be the square root, hopefully. Anyway, I don't know is the simple answer. And it's a really important problem. It's a really important question. And the only possible avenue for getting past it is a very open, forthright public engagement.
When I was 15 years old, I got dengue, and I was in bed for a week. It was really quite something. My other connection with Ethan's work is that I actually, in 2000, when I started working at Becton Dickinson, we did a bit of a collaboration project for a couple of years together. My dissertation was also on Drosophila genetics, a long time ago at UCLA. Do we have any questions on that side of the audience? No? Yeah, go ahead. Mike, can you help me with the microphone? Sure. Thank you.
Hello. Hi. So my question is a little less specific, but you touched on applications, possible applications of MCR and active genetics to cell therapy, tissue regeneration therapy, and those medical human health aspects. So for me, could you perhaps elaborate on possible ways in which you could apply this technology, not like a gene drive, but to those applications like tissue regeneration or other avenues?
Yeah, so what we're aiming to do is using the extremely high efficiency of copying that we have observed and trying to bring that principle from the germline of insects to human cells, which might seem a very big jump, but the very basic mechanism that repairs the DNA after a cut is extremely well conserved. And so we want to bring that kind of knowledge into human cells and apply it to bring in very large pieces of DNA, which right now with the current technology, even if it happens very efficiently, it's not, the pieces of DNA you can bring in are very small, or they have a limit, which is whatever you can package into a virus. And we believe that we can bring in with active genetics not only like larger pieces of DNA, but also get them on both alleles, both copies of the chromosome, and therefore just really could open like a whole new era of synthetic biology into human cells. And you can bring in like whatever feature you want. And if you look like down the line, maybe 5 or 7 years, how this could be applied is that you could not just modify one specific locus, but like 10 different locus at the same time in one cell and generate whatever cell that does whatever you want the cell to do and not what it's programmed to do normally.
Have you guys seen the movie Gattaca? Who've seen that movie?
Yeah, I always liked it.
Okay, so this is a technology that could potentially make Gattaca a reality, right? This is fascinating, but again, I feel like it's just a teaser. So obviously, this is a really, really important transformative technology that is at a very, very early stage. And the question, that I was asking Ethan and Valentino is, how are we going to make this, how are we going to transform this into something that has the impact and the potential that it can do?